Hello, my name is Rodrigo, and today I'll be talking about high content screening. High content screening, or HCS, is essentially an extension of quantitative microscopy to large scale experiments, measuring hundreds or thousands of conditions or perturbations at once. Instead of focusing on a single readout like cell viability, HCS involves capturing rich, image based information about cells, including their shape, structure, internal organization, and more. A key feature of many HCS experiments is the use of multiplexed imaging approaches, often referred to as cell painting. In these assays, cells are labeled with a combination of dyes or fluorescent reporters that highlight different cellular features all at once. In this example on the right, you can see 5 different dyes used to visualize different cellular compartments, including the mitochondria in red, the cytoskeleton in orange, and the nuclei in cyan blue. This is then paired with automated imaging, allowing us to get a host of measurements simultaneously. At a high level, high-content automated imaging captures a high-dimensional snapshot of cellular behavior for thousands of cells, which are then used with screening libraries to enable large-scale candidate selection. Next, let's talk about HCS experimental design. These experiments are usually performed in multi-well plates, where each well contains cells exposed to a different condition. This could be a different drug, genetic perturbations like using CRISPR knockouts, or some environmental change. After treatment, in some cases, cells can be imaged live, but most commonly the cells are fixed and stained with fluorescent labels, and then automated microscopes capture images across all wells in multiple fluorescence channels, and sometimes across multiple focal planes as well, creating what is known as a Z-stack. The result is a large collection of images, essentially a data set of cellular snapshots under many different conditions. As I mentioned before, the key idea is that images contain far more information than a single measurement. What makes HCS really powerful, though, is its flexibility and potential for customization. In this example, researchers use a CRISPR library to generate knockouts, but what is truly unique here is that for an HCS experiment, what is not very common is here they generate cell libraries where each cell has a unique perturbation, and these cells are pulled together before phenotypic images are acquired, showing how there is flexibility in the workflow, and that is without going into the options for labeling and imaging. Any number of phenotypes can be studied using different combinations of dyes or image features. When you consider these, the possibilities for assaying even highly complex phenotypes are truly endless. That being said, let's talk about some types of questions HCS enables. First, we can ask what compounds or genes affect the phenotype of interest. This is the most direct application. If we have a specific phenotype in mind, like changes in cell shape, organization, or a disease-related feature, we can screen many genetic or chemical perturbations and identify which ones produce that effect. This allows us to systematically link genes or compounds to specific cellular outcomes. Second, we can ask, how do different compounds affect cells across many morphological features? Instead of reducing everything to a single measurement, high-content screening captures a rich set of features simultaneously. Going back to this image on the bottom right, highlighting different parts of the cell, when testing many drugs, one compound may affect mitochondrial number, one might affect nuclear size, while another may affect both of those features and others all at once. Rather than missing those effects, we can quantify changes across all these features and build a more complete picture of how the cell is responding. Third, we can ask, which perturbations produce similar morphological profiles, and what does that reveal about mechanisms? Even if two perturbations are very different, if they produce similar cellular phenotypes, that can suggest that they're acting through the same pathway or biological process. In this example on the top right, each point represents a different genetic perturbation. You can see that several genes cluster together, indicating they produce similar morphological effects. In this case, those genes are all related to mitochondrial translation, showing how phenotype similarity could reveal functional relationships as well. Finally, with the widespread use of AI, one big type of question we can ask is, can we teach models what phenotypes of interest look like and use that to discover new candidates? Here, instead of manually defining what we're looking for by somehow translating our phenotype to a quantitative measurement, we can train machine learning models to do this for us and recognize complex phenotypes, either from images directly or from extracted features. Once trained, these models can scan large data sets and identify new perturbations that match the phenotype, enabling even more automated and scalable discovery. Thank you for watching, and thank you to the University of Arizona's Data Science Institute, the Colleges of Science and Information Science, and the university libraries for making this video possible.